of Washington's Engage program, here to welcome you to our event tonight. My research focuses on figuring out where the instructions for traits like drug resistance are located in the yeast genome. And I get to have this opportunity because of the special collaboration between Town Hall and the UW's Engage program. Together, they host a series called UW Science Now as part of the Seattle Science Lectures. Our speakers program will run about 25 minutes, including 5 minutes for your questions. We ask that you come to the mics at the edge of the stage so we can pick you up for the recording that we're making. Before I introduce our first guest, though, I'd like to mention some other events from our science series. First of all, please join us tonight for a panel discussion with researchers from the ISB on the future of health tonight at 7:30, following these Engage talks. Also, the next 2 talks on the UW Science Now series will be right here on April 6th, starting at 5:30 p.m. Siva Kasinathan, Jonica Ash, and I will talk about our work, which spans genomics and infant mental health. And now, for our first guest, Alison Weber. Allie Weber is a graduate student at the University of Washington studying the neuroscience of vision. Today, she's going to tell us about her work studying the neural codes that govern visual adaptation. Please give a warm welcome to Allie Weber.
Hi, can everyone hear me?
Yes.
Great. Okay. Thank you, Anne, for the introduction. Thanks, everyone, for being here. Thanks to Town Hall for having us. So today I'm going to tell you about my work deciphering the code of the retina. So first I'm going to tell you a little bit about the retina and what I mean by deciphering its code. Then I'm going to tell you how deciphering that code might help us to restore sight to people who have lost it. And then finally I'll tell you a little bit more about my work specifically at the University of Washington. But first, I want you to imagine that you're taking a hike through the mountains. The sun has started to set, and you decide to sit down and enjoy the beautiful scene in front of you. You're looking down at the trees down in the valley, the beautiful colors in the sunset, the brightly lit rocks in front of you, and you think to yourself, I better get a picture for my Instagram. And so you take out your camera, but unfortunately your picture ends up looking something like this. That's probably not what you had hoped for. I think we've all had this experience where you snap a picture and it just doesn't capture the original. So we've lost all the detail of those trees down in the valley, the hills in the distance. All you get is this bright spot of the sun, maybe a little of the rocks in front. But rather than focus on all the problems that your camera has, what I'm interested in is how your eye is able to do such a good job in the first place. So how is your eye able to do this when a camera cannot? How is it that you can see all the colors of the sunset, those trees down in the deep shadowy valley, and the brightly lit rocks in front of you? How is your eye able to deal with that huge range of brightness all in the same scene? When I first started thinking about this question, I thought that maybe the pupil had a lot to do with it. So the pupil is that black dot in the front of your eye. If we look at it from this view, this is as if we cut the eye in half and are looking towards the inside. And it's that opening in the front of your eye that can expand and contract to let different amounts of light in. And that is what the pupil is there for, to help your eye deal with different light levels. But how much does it actually help? Well, let's take this bar to represent the full range of light levels that you can see. So down at the very darkest level, it actually turns out that humans are able to detect a single photon. So that's like being able to see the flame of a single candle from several miles away. It's incredibly dim. Okay, then at the upper range, the brightest conditions under which you can see are 1 billion times brighter than that. So this is an incredibly huge range that your eye is able to deal with. Now, how much does the pupil help? Well, it turns out just a little bit. So the amount I've shrunk it by is the amount that your pupil actually helps. And so that means that the range that's left here, some other part of your eye has to deal with it. And that some other part of the eye is your retina. The retina is the thin sheet of cells at the back of your eye that detects light. And that's where vision starts. So let's take a minute to zoom in on the retina. So the image I'm showing here is actually a mouse retina, which is what I study, but it's actually very similar to human retina, and we'll come back to that in a few moments. Now, it's artificially colored just so that we can see the different cells more easily. But I hope that the first thing that stands out to you is that it's very neatly organized into these nice layers. So let's step through these layers. Starting in the very back of your eye, those are the rods and the cones. Those are the cells that actually detect the light. And these are the ones that Rachel is going to be talking about in the next talk, so keep these in mind for later. Now, if you're thinking to yourself, wait a minute, this doesn't make any sense. Why are the rods and the cones all the way in the back of the eye if they're the ones detecting the light? That is a reasonable question. And it turns out that this is actually just sort of a strange quirk of evolution, that the cells that detect light are at the very back of our eye. And that's true for us and for all other mammals. There are some other animals, like actually the octopus eye, has their cells arranged so that the cells that detect light are the first ones that the light hits, but that's not how it works for us and other mammals. Okay, so once those rods and cones detect light, they pass that message on to the next layer of cells, and that's called the bipolar cells. And you can think of those as sort of relay cells that will send the message on to the next layer. And the final layer are the ganglion cells, and these are the ones that I study. And we call these the output cells of the retina because they're the ones that send that message on to the brain. Now, I mentioned that the human and the mouse retina are actually quite similar. So let's look at them in comparison. So one of these is human, one of these is mouse. They look different than the last slide just because we've used a different method to color the cells. But I hope you can still see that there's this neatly layered structure in both of them, and they're really quite similar. They both have these three nice layers. And even to my eye, as someone who studies the retina, these look incredibly similar. And so we think that a lot of the insights that we gain from studying the mouse will actually help us to learn about the human retina as well. So it turns out that the one on top is the human and the one on bottom is the mouse. But what I want you to take away is just how similar they really are. So I keep telling you that once the rods and cones detect light, they send that message on to the next layer. Then the message moves through that layer onto the next. But how do these messages actually move through each layer? So to think about that, let's focus in on one of these ganglion cells. And from now on, I'm just going to draw my ganglion cell like this for simplicity. And the cells in your retina are actually neurons. Neurons are the cells that make up your brain and the rest of your nervous system. And what makes neurons special is that they are electrically active. And so when one of these cells gets an incoming message from another neuron, it triggers a tiny little jolt of electricity, or what we call a spike or action potential. And that travels down the length of the cell and allows the message to be sent through that layer. Now these tiny spikes, like I said, they're very small. So the average size of one of these is only about 100 millivolts. So if you think about the electricity in your house is 120 volts. These are incredibly teeny tiny little jolts of electricity. And the way that these spikes convey messages is that a pattern of spikes will send a particular message. So a lot like patterns of dots and dashes in Morse code spell out letters and then words, the patterns of these spikes send a message to the brain about the light that is coming into the eye. So a pattern like this might signify that there's flickering light. And I just want you to remember that I told you that the ganglion cells are the ones that send their messages on to the brain. They're the only cells from the retina that send messages on to the brain. And so that means that everything you see in the world starts in this form, as this electrical activity. Now, I promised to tell you how understanding this code will help us to restore sight to those who have lost it. Many forms of blindness start due to damage to the retina. And one particularly common kind of damage is damage to the rods and cones, those cells that detect light. And so if those cells aren't detecting light, they have no message to send on to the next layer. There's no message coming into the ganglion cells, and they have no message to send on to the brain. But is there some way that we can send the right messages to the ganglion cell anyway? Scientists are currently working on devices that we can implant into the retina that can electrically stimulate those cells. And so if we know what those patterns of electrical activity are supposed to look like, we can mimic those and send the right signals onto the ganglion cells anyway. And so if we want to send the right messages, we need to understand that code. And in order to crack the code, we need to listen in on the messages that are being sent in the healthy retina. So at the risk of sounding a bit like the NSA, we're going to listen in or intercept the messages that these retinal neurons are sending. So it's a little bit like holding up a tiny microphone to the cell. We want to amplify those teeny tiny electrical signals. But since the signals are electricity, not sound, we don't use a microphone. We use what's called an electrode, and that allows us to record these patterns of electrical activity inside the cells. And now I want you to think back to the picture that I showed you at the beginning, and I told you that I'm interested in how are you able to see those dark, shadowy trees down in the valley, but also the brightly lit rocks in front of you. And so I want to know what these signals look like. What does the code look like when either there's a lot of light coming into the eye or very little light coming into the eye? And so I can record from these cells in both of those conditions. So I shine a lot of light on the retina and record those electrical signals, or I let the retina sit in the dark and record those electrical signals. And so hopefully what you'll notice here is that for the cells that I record from in the dark, there are very few of these spikes. Compared to when I'm shining a lot of light on the retina, there are a lot of these spikes. And so this is sort of the first feature of that neural code. For these cells, more spikes means more light. Okay, but when you're looking at that beautiful sunset, you don't just stare down into the valley for several minutes, and then stare up at the rocks for several minutes, and then stare at the sun for several minutes, but you're constantly looking around the scene. So I don't really want to just know what happens when the retina is sitting in the dark, or when I shine a bunch of light on it, but what happens when you quickly switch from one of those conditions to the other. So to test this, I first start with the retina sitting in the dark, and then I suddenly switch on a light and record the signals there. And so what you'll see is that when we first start off with the retina in the dark, like we saw before, there are very few of these spikes. And then as soon as the light comes on, the cells fire a whole bunch of these spikes. And this makes sense based on what we saw before. This is part of the code, where more spikes convey that there's more light coming in. But if we look at another window here, that's also when the light comes on, you'll notice that there are many fewer spikes in this window. So even though the same amount of light is coming in, the messages that these cells are sending out are different. Okay, but I want to make sure this doesn't just happen once. I'm a scientist, so I do this over and over and over and over again. And so here I'm showing you 10 examples, but in reality I do this more like 100 or 1,000 times. And then if I look at what happens on average, I see that what we saw in the first example is borne out. So when the cells are sitting in darkness, this firing rate, or the rate at which those spikes are happening, is very low. And then as soon as the light comes on, a whole bunch of spikes are emitted. And the rate at which those spikes are happening it peaks at about 250 spikes per second. So think about that. As you're looking around the room now, the cells in your eye are firing hundreds of times per second. It's incredibly fast. Now, as that light continues to shine on the retina, the firing rate starts to come down, and we call this process adaptation. So even though the exact same amount of light is coming in, the rate at which these spikes are being sent out is different. So the message that the brain is getting is different in each of these cases. So why might that be happening? Why should the retina be doing that? Well, to understand this concept of adaptation, for a moment, let's forget about messages and codes, and let's think about something completely different. Imagine that I've asked you to help score a local gymnastics competition at an elementary school. Okay, so you'll be scoring elementary school students, and I tell you that you can give them a score of 1 through 10. And maybe you don't give anyone a 1 because you don't want to make any of the young students feel bad. But some people get higher scores, some kids get lower scores. You sort of use the range of scores. And so you're going along enjoying this competition, and now imagine midway through the competition, all of a sudden, unbeknownst to you, I swap out all of those gymnasts from your local elementary school and I replaced them with Olympic gymnasts. So suddenly they're all much better. But I didn't tell you that I was doing this. Okay, so if you're using your old scoring system, you're going to see someone, the first Olympic gymnast, and say, wow, that was really amazing. That's definitely a 10. Okay, the next one comes up, also an Olympic gymnast, unbeknownst to you. And you think, whoa, what are the odds? Two such incredibly talented gymnasts right in our backyard. Okay, so maybe after a few of these you would start to get a little skeptical. Maybe you would realize that something about the competition had changed. And maybe your old scoring system doesn't make quite as much sense in these new circumstances. So over time, you'd sort of adjust your scoring rubric. You might make it a little more difficult so that you could go back to using a range of scores to judge these competitors, so that you could actually compare their performance instead of just giving everyone for the rest of the competition a perfect 10. This is actually a lot like what's going on in the retina when adaptation is happening. So you can imagine that when the retina is sitting in the dark, it's set one scoring system for itself. And as soon as that light comes on, for a moment it's using its old scoring system. And so it signals that this is an incredibly bright light. It gives basically the highest score possible. It's like saying perfect 10. It fires a bunch of these spikes. And this happens for a little bit, but eventually the retina starts to adapt. It starts adjusting its scoring system, and so that the rate at which those spikes are emitted comes down. And so let's imagine for a second what would happen if this adaptation didn't occur. That means that when you are looking down into the valley, and then you suddenly looked up at those bright rocks in front of you, your cells would just fire away like crazy the whole time. So it would be sending the maximum signal the whole time. They would be saturated. And so they wouldn't be able to send any messages about the small little dark flecks in the rock or the subtle shadows that help you to determine the shape of the rock. And so changing the code actually helps the retina to convey more information and allows you to see those details in the scene over a bigger range of light levels. There are a lot of features of adaptation that we still don't understand, and one that I'm particularly interested in is how does the retina determine when to change its code? So if you think to the gymnastics competition, how many of those Olympic gymnasts would you have to see before you realized that something had changed and you needed to adjust your scoring system? And one of the interesting things about the retina that we know is that the rate at which it adapts, or when it decides to change its code, is actually different in different circumstances. And so that's something that I'm studying in my ongoing work at the University of Washington. So I hope that you might keep some of this in mind, and next time maybe that you're enjoying a beautiful sunset, the thought might cross your mind that the cells in your eye are sending messages onto your brain at this incredibly fast rate, and they're constantly changing and adapting the code that they're using to send those messages. And all of that is what allows you to enjoy the scene in front of you. With that, I want to thank Town Hall for hosting us here, the Engage program for making all of this happen, the labs that I'm in at the University of Washington, the Riki and Shea Brown labs, which I'm happy to see some familiar faces from in the audience. And then we have a few questions, I think we have a few minutes for questions now, but also feel free to contact me and ask any questions. I'd be happy to have them. Thank you. I think for questions, please come up to the microphones at the side of the platform.
Thank you.
Yeah.
With that initial spike in the adaptation, is it contrast that the retina is responding to?
Yeah, so the adaptation can be affected by both changes in brightness and also what we would say are changes in contrast. So yeah, that's one of the things that's part of my ongoing work, where I'm trying to determine exactly which parts of a scene are actually changing that rate at which adaptation occurs, and contrast is one of those.
Have you done any research on the middle layer? Does that have any impact on the adaptation?
So it turns, yeah, that's an interesting question. So there's adaptation happening at each of those layers. There's also adaptation happening in the rods and cones, there's adaptation happening in the bipolar cells, and there's also adaptation happening in the ganglion cells. And yeah, I haven't studied that personally, so I can't tell you any of the specifics of it. But it seems to be a general phenomenon that happens also not just in the retina, but in other parts of the brain as well. So in the auditory areas that help you hear, there's also adaptation. It happens basically throughout the brain, so it's a very general phenomenon. Hi.
Thank you so much for the great talk. I was just curious to know why, when you see the adaptation, the firing rate never goes back down to baseline the way it used to be?
Yeah, that's a good question. So our thinking on that right now is that the fact that the baseline rate stays higher is sending a signal that the average light level, or the light that's coming in, is in fact brighter. So sort of the average rate when it's dark is lower than the average rate when it's bright. The adaptation just keeps it from completely saturating, but you'll still have a sort of a baseline or steady state higher firing rate when there's a higher light level.